So our remote lab this week has two parts. The first is to explore LeChatelier's principle, and the second is to determine the value of Kc for a particular reaction. So in the first part, we will explore LeChatelier's principle together. And so as a reminder, LeChatelier's principle says that if a system at equilibrium is stressed, which remember, stressed means changed in some way, then the system will shift, which means react, in order to counteract that stress and return to equilibrium. We are going to consider two types of stress. We're going to focus on concentration, so adding or removing reactants and products, and also on temperature, so adding or removing heat from our equilibrium systems. So the module that you'll work with will guide you through a series of observations. I want you to talk about these observations with your team and connect each of those observations to some sort of chemical reaction. So it'll be a chemical reaction that is at equilibrium or could be at equilibrium to chemical reaction. And then, more importantly, I want you to explain every observation using LeChatelier's principle. So as you're talking, think about how does LeChatelier's principle apply here? At the end of this part, sort of at the end of the whole day, what you need from this part is a team document. So your post-lab is a team document that explains your observations. So appoint someone to turn that in, but work on it together. Okay, so there's part one. Some qualitative observations, explain how LeChatelier's principle applies. In the second part of the lab, we're going to actually determine the value of Kc. And so maybe you haven't really thought about this. How do we actually use experiments to figure out what the value of Kc is? And so the answer is that we're going to prepare equilibrium mixtures, measure their concentrations, and then calculate K. The reaction that we care about is iron 3 plus thiocyanate, SCN, becomes iron thiocyanate. And so we could write the Kc expression using the concentrations of the reactants and the products. And so if we knew the equilibrium concentrations of those things, we could find Kc. And so normally if you were going to calculate equilibrium concentrations, you would know some sort of initial molarity for each of your reactants. And we're going to start with no product. You could define how much the change would be, and then you would calculate the equilibrium amounts using your value of the equilibrium constant. In this case, we don't know the value of the equilibrium constant, so instead we're going to take advantage of the fact that iron thiocyanate, FeSCN, is red. And so we're going to measure its absorbance in order to determine the concentration. So once we know the concentration of FeSCN at equilibrium, that means we know the value of X. And now we can back calculate to find the equilibrium concentrations of the other components. So how are we going to know the initial molarities of our two components? These are things that we can fix or determine by how we set up the experiment. So you can know the concentrations of iron that you make initially, and you can know the concentration of FeSCN, and you can calculate what their concentration would be initially once you mix them. And so we can know what those are. We can decide what they are in our experiment. In this experiment, we're also going to make them fairly similar. So they might differ. They're going to differ a little bit, and we're going to try several different mixtures, but they're going to be kind of on the same order of magnitude. We're going to repeat for several mixtures, and then average to find the average. And so what's nice about the average is that it will help to cancel out some of the random errors that might occur in each individual measurement. Okay, but so we're going to measure the absorbance of the iron thiocyanate. But how do we know the concentration if we have the absorbance? Before, we've talked about Beer's law. And Beer's law tells us that there is a direct relationship between absorbance, A, and concentration at a specific wavelength. So that absorbance is proportional to concentration at a specific wavelength. Okay, so what we can do, what we need to do is find epsilon and B. So we said, so epsilon is the molar absorptivity, and it's a proportionality constant that relates concentration and absorbance. B is the path length. And so that is how, basically the width of the cuvette. We know that that is 1 centimeter for our spectrometers, but that's still sort of part of our equation. So the way we're going to find the value of the molar absorptivity so that we can relate concentration and absorbance is first we're going to prepare solutions of known concentration. And in this case, that'll be known concentrations of iron thiocyanate. And then we'll measure the absorbance. So we'll know the concentration and we'll know the absorbance. So what wavelength should we use to measure the absorbance? Well, in general, we always want to use the wavelength of wherever the peak is, because that's the place where we're going to see the biggest changes in absorbance as we change concentration. And so for this solution, that's going to be at about 450 nanometers. And so then when we have measured the absorbance at 450 nanometers, we can plot the absorbance versus the known iron thiocyanate concentration. And so we'll make another graph where we have absorbance at 450 on one axis, and we'll have concentration of FeSCN2+ on the other axis, and you'll plot your points and fit a line using linear regression. And so that line, y equals mx plus b, so the m, the slope, will be your epsilon b, and the intercept will be close to 0, but not totally 0. So we'll use the equation of that line in order to calculate concentration from known absorbances. Okay, wait a minute. So all of this was predicated on knowing the iron thiocyanate concentration. But this system is in equilibrium, and we don't know the iron thiocyanate concentration if we mix together known concentrations. Wasn't that the whole point of the experiment? You're right. This is a problem. How are we going to know the iron thiocyanate concentration if we don't know Kc in the first place? The answer here is that we're actually going to make use of LeChatelier's principle. So if we have iron 3+ and we add iron thiocyanate, we're going to make FeSCN, the iron thiocyanate complex. But if we add a very large excess of thiocyanate, then that will drive the reaction more and more and more toward the products. So basically we keep increasing the SCN concentration, and so that will push the equilibrium toward the product side. So basically what happens is we drive this reaction to completion so that iron 3+ becomes the limiting reactant. And then, since there is a 1-to-1 molar ratio between iron 3+ and FeSCN, we find that the FeSCN concentration in the mixture equals the concentration of iron that was added. Now, we will need to consider that when we mix these together, we need to consider any effects of dilution. And remember that you can calculate dilution from M1V1 equals M2V2. Okay, so we're actually going to reverse the order that we just talked about. The first thing that you're going to do in this experiment is make that Beer's law calibration curve. And so the module will guide you through thinking about how those solutions are prepared, and it will give you data for the absorbance that you would have measured based on concentration. And then you're going to measure the absorbance of some equilibrium mixtures, or rather you're going to get the data related to measuring the absorbance of those equilibrium mixtures. So we'll do what we talked about last first, and then work our way back to where we're basically filling in X in our ICE table. You'll use that ICE table concept in Excel to calculate Kc, and then compare to a published, or what we would call literature, value. So this was an experiment that somebody else did, and your job is to think about, is your number the same? Is it different? Why might there be differences? What might be the sources of systematic and random error that would cause differences between your experiment and theirs? Okay, so once you've done all of that, you are responsible for coming up with an individual abstract and turning in an individual spreadsheet. You're certainly encouraged to talk to your team, talk to your TAs while you're completing this experiment, but at the end of the day, you're responsible for your own work. So remember, post your questions to Slack and answer the pre-lab questions in your notebook, and have fun with this. This is a pretty cool way where you can see the experimental applications of equilibrium.